Hello friends, in this section we are going to focus on process of implantation of embryo in human, especially in three parts as in vitro fertilization embryo transfer, gamete intrafallopian transfer and zygote intrafallopian transfer. But prior to understand these processes, it is mandatory to understand the process involved in fertilization. What we are going to cover in this lecture is looking at the events of fertilization, and then we will talk about early embryonic development and implantation as well as embryonic and fetal development. Fertilization. Fertilization usually takes place in the uterine tube. As we have known, sperm would have to be introduced into the female reproductive tract within 5 days before ovulation. Remember, time is everything. The sperm are viable for 5 days and the secondary oocyte is viable for 12 to 24 hours. So, the sperm actually they are initially incapable to fertilize, and then later on they become capable of fertilization through a process called capacitation. So, what capacitation is? It's where actually the tail or a flagellum of the sperm moves faster, so that actually it increases motility and the plasma membrane of the sperm is altered, and it's altered in such a manner that it can fuse with the secondary oocyte. This is just a sort of pictorial view of what we are just talking about. Now let us learn how secondary oocyte production takes place inside the ovary and how the eggs are produced inside a human female reproductive system. So, while it is traveling through the uterine tube, it may encounter sperm. So, the sperm have to enter through the female reproductive tract and the sperm have to encounter the secondary oocyte, and that is where fertilization takes place. Thus, fertilization takes place in the uterine tube. Remember that implantation takes place in the uterus. Now, let us understand the process to fertilize the ovum and the penetration of the secondary oocyte. How does the sperm penetrate? So, there is a whole bunch of sperm that actually reach the ovum. Remember that for every ejaculation, there is anywhere from 50 to 500 million sperm that get released. So, that means a whole bunch of sperm can actually reach the ovum. Early stages of embryonic development. As we have studied that the sperm enters the oocyte to conceive and fertilize an egg cell for a resultant zygote formation, the role in the human body begins with a single cell. It begins to divide in roughly 30 hours after fertilization. It divides into 2 cells, then 4, and so on without really gaining any extra size. So, after a few divisions, we are left with a big ball of cells that's referred to as a morula. The cells keep dividing then and eventually run to get nutrition from the surface. So, there is a cavity that forms inside, and that's going to be called the blastocyst cavity, and we will take a look at that to get a cross section of the morula. So, we have cells surrounding in it, then within it, that as it gets larger and larger, the cells divide. That blastocyst cavity that forms to allow nutrition to get to and from these dividing cells. So, at this point, the morula is known as blastocyst, and this is the blastocyst cavity. Thus, division keeps happening and the blastocyst gets larger and larger, and one group of cells goes to sequester after one end of the blastocyst. On the other end, cell mass is going to form the placenta and other related structures. We have cells. They are still in contact with the blastocyst cavity, but the cell are going to sequester away from it, and these cells are known as epiblast. The cells that are near to blastocyst cavity are known as hypoblast cells. Now, important thing about these epiblast cells is that these will form the actual embryo. As we have discussed before, these are the cells that are actually going to form really all the structures of the developing human. 3 cell layers form our actual mature body, and the first thing that happens is a little strip that appears on the one side referred to primitive streak, and at the very tip that, as we recall, the primitive pit. An important region called the primitive node is the place for gastrulation that happens, and it's very difficult to visualize this. We have the epiblast disc balls and the hypoblast disc balls that really contribute to the mature embryo. This then forms the endoderm and the mesoderm, which is at the epiblast cell layer on top, which is now magical referred to as the ectoderm. In red, we have the mesoderm, and in yellow, we have endoderm. The term is not the same as the hypoblast. All these layers come from the epiblast cells, so ectoderm in blue, mesoderm in red, and endoderm in yellow. Process of implantation of embryo and placenta formation. The implantation process is the stage that occurs and should occur immediately after conception. Conception is the process that is being achieved by a successful early developmental stage of formation of zygote and the resultant immature embryo stage whilst development. Implantation is rather a natural process of development that establishes an embryo inside and attached to the walls of uterine tube, which then adheres and derives nutrition and blood from the recipient's or mother's body. After traveling down the uterine tube, the early embryonic developments itself into the inner wall of the mother's uterus. This process, called implantation, begins 6 days and ends 10 to 12 days after fertilization. Growing embryo begin to produce a hormone called human chorionic gonadotropin, referred to as HCG, the substance detected by pregnancy test. HCG directs maternal hormones to interrupt the normal menstrual cycle, allowing pregnancy to continue. Techniques followed for successful implantation. The process of implantation of the embryo for the successful assisted placentation could be achieved more or less by involvement of IVFET, GIFT or ZIFT. Let us have information for the individual techniques used. In vitro fertilization and embryo transfer. The field of reproductive medicine has changed forever with the birth of Louis Brown in 1978 by in vitro fertilization and embryo transfer. Patrick Stiptoy and Robert Edwards of England are remembered for their revolutionary work. The past decade has witnessed 2 more dramatic changes in the technique protocol of in vitro fertilization and embryo transfer, IVFET. One such change was from natural cycle to superovulation protocol, and the other one was replacement of laparoscopy by vaginal sonography for ovum retrieval. The indications of IVF are used in the tubal disease, unexplained infertility, endometriosis, male factor infertility, multiple factors as found in female and male, failed ovulation induction, ovarian failure as in donor oocyte IVF, women with normal ovaries but no functional uterus, referred to as Mullerian agenesis, and last, women with genetic risk as found in IVF and PGD. Selections of patient criteria includes age less than 35 years, presence of ovarian reserve, husband normal seminograph, couple must be screened negative for HIV and hepatitis, normal uterine cavity as evaluated by hysteroscopy or sonohysterography. Principal steps of an ART cycle includes downregulation using GnRH, agonist controlled ovarian stimulation, monitoring of follicular growth oocyte retrieval, fertilization in vitro as in IVF, ICSI and GIFT, transfer of gametes or embryos, and the last, luteal support with progesterone. Embryo transfer. The fertilized ova at the 6 to 8 blastoma stage are placed into the uterine cavity close to the fundus about 3 days after fertilization through a fine, flexible, soft catheter transcervically. Not more than 3 embryos are transferred per cycle to minimize multiple pregnancies. The process of transfer should be accurate, atraumatic and aseptic. Small volume transfer using soft catheter under ultrasound guidance gives the best result. Trial transfer is beneficial. The number of embryos to be transferred depends mainly on maternal age and the embryo quality. Excess oocytes and embryos can be cryopreserved for future use. This will reduce the cost of ovarian hyperstimulation. Luteal phase support is maintained with progesterone. It is started on the day after oocyte retrieval. HCG is given in supplemental dosages, for example 1500 to 2500 international unit. Micronized progesterone 200 mg thrice a day oral or as vaginal suppository or progesterone in oil injection 50 mg IM daily is continued for about 14 days. By this time, diagnosis of pregnancy by estimation of beta HCG is possible. There is increased risk of miscarriage about 18%, multiple pregnancies about 31%, ectopic about 0.9%, low birth weight baby and prematurity. The risk of congenital malformation of the baby remains similar to general population. Prognostic factors for IVF-AT. Maternal age. There is age-related decline in response to ovarian stimulation, less oocytes, poor oocyte quality, less embryos and implantation rate. Ovarian reserve declines with age. Indication of IVF and past reproductive success. Women with tubal or ovulatory factors, endometriosis or unexplained factor have higher success rate compared to women with poor ovarian reserve. Presence of hydrosalpinges affects the outcome adversely. Fibroid uterus, especially the submucous or interstitial variety, have adverse outcome. And the last, smoking, poor outcomes are observed phenomenon. Gamete intrafallopian transfer. Gamete intrafallopian transfer was first described by Esch and colleagues in 1984. It is a more invasive and expensive procedure than IVF, but the result seems better than IVF. In this procedure, both the sperm and the unfertilized oocytes are transferred into the fallopian tubes. Fertilization is then achieved in vivo. The prerequisite for GIFT procedure is to have normal uterine tubes. The indications are the same as that of IVF except the tubal factor. Best result is obtained in unexpected infertility and the result is poor in male factor abnormality. The superovulation is done as in IVF. 2 collected oocytes along with approximately 2 lakh to 5 lakh motile sperms for each fallopian tube are placed in plastic tube container. It is then passed through the laparoscope and inserted 4 cm into the distal end of the fallopian tube where the combination is injected. The overall success and result in delivered pregnancy rate is as high as 27 to 30%. Zygote intrafallopian transfer. Zygote intrafallopian transfer was first described by DeVry and colleagues in 1986. The placement of the zygote following 1 day of in vitro fertilization into the fallopian tube can be done either through the abnormal ostium by laparoscope or through the uterine ostium under ultrasonic guidance. This technique is suitable alternative of GIFT when defect lies in the male factor or in the cases of failed GIFT. Results and success rate is as good as that of the GIFT, approximately 29 to 30%, or ZIFT is avoided when tubal factor for infertility are present. The risk of ectopic pregnancy is high for GIFT and ZIFT compared to IVF. Ethics, hazards of implantation and safety measures. Opinions on the moral status of the human embryo remain deeply divided. For those who believe that the embryo has equivalent moral status to a grown human person, research involving the use or destruction of human embryos is likely to be unacceptable or acceptable only in a limited range of circumstances. Yet, this was not at the heart of the objections raised over this study. Many scientists and many jurisdictions accept human embryo research, notably in the context of stem cells. The issue in this case was not the use of embryos in general, but their use in particular type of research. Only embryos donated with ethics committee approval and patient informed consent can be used for research. Couples can have their choices for embryos. They can continue to keep them cryopreserved, then they can have them discarded, or they can donate them either for reproductive purposes to other couples or for research purposes, provided that an embryo protocol or bank is willing to accept the embryos. The creation of embryos for research rarely occurs and is not consistent with policy in most countries. In summary, the majority of human embryos used for research are surplus to the treatment of infertility and would otherwise be discarded. Implantation failure is considered to be caused by inadequate uterine receptivity in 2/3 of cases and by problems with the embryo itself in the other third. Inadequate uterine receptivity may be caused by abnormal cytokine and hormonal signaling as well as epigenetic alterations. Recurrent implantation failure is a cause of female infertility. Therefore, pregnancy rates can be improved by optimizing endometrial receptivity for implantation. Evaluation of implantation markers may help to predict pregnancy outcome and detect occult implantation deficiency. Luteal support is the administration of medication, general progestins, for the purpose of increasing the success rate of implantation and early embryogenesis, thereby complementing the function of corpus luteum. However, in women with more than 3 implantation failures in assisted reproduction, a review of several small randomized controlled studies estimated that the use of adjunct low molecular weight heparin improves live birth rate by approximately 80%. Summary. Friends, now it's a time to summarize what we learned today. We have learned the process of fertilization and early stages of embryonic development in detail. We have gone through the various types of embryo transfer, namely IVFET, GIFT and ZIFT. Lastly, we have also discussed about the ethics, ethical issues, hazards of implantation and finally the safety measures as well. Implantation of the human embryo is complex interaction between the endometrium and the mobile blastocyst. It must occur within a relatively narrow time frame under conditions of a primed receptive surface, epithelium and a morphologically changing trophectoderm. Mechanically, the movement of the blastocyst must be arrested in order for attachment and then invasion can occur. Normal implantation is crucial for successful pregnancy. For the infertile couple and their treating physician, a better understanding of the processes of implantation will enable better diagnosis and treatments to overcome the reasons why they cannot have a healthy child. Together, by selecting of the healthiest embryo and establishing the most receptive endometrium, we can increase implantation efficiency. Future research into markers of endometrial receptivity will allow clinicians to define the optimal environment in which to transfer the best embryos, thereby improving pregnancy rates and decreasing complications such as multiple pregnancies, miscarriages and ultimately conditions that compromise the healthy intrauterine development of babies. Thanks.